I would like to welcome every one of you to the Alumni Leadership Conference. As some of you might know, my name is Nicolas Chamas, SM87, and this year I am serving as your proud Alumni Association President. I have traveled here from my home country of Lebanon to once again connect with my fellow MIT alumni and volunteers for the Institute. You have also traveled from near and far because we share a passion for how MIT is making a better world, not only for us, but for generations to come. Over the next 2 days, you'll have the opportunity to connect with one another, with current students, with fabulous faculty, and with the staff teams in the AA and resource development. I know you will be inspired to continue to engage in the important work you do as a volunteer for MIT. By the end of the conference, it is my goal that you will feel energized, excited, and even more motivated than you are today. I also hope that you will take what you learn here and share the stories of MIT when you return home. Along the way, I hope you will write down a few things you can do to advance the mission of the Institute, whether that's inviting a friend to get involved by coming to an event, or making an annual fund gift, or also by sharing how MIT makes a difference in the world with prospective students or friends of the Institute. I hope you have already downloaded the MIT Alumni Events app from Guidebooks. If you haven't done so, please go to the App Store or Google Play to download it now. It is the best way to make connections with your fellow attendees, get alerts and updates, and manage your own conference schedule. If you need help, please see one of our staff in the registration area. Today, we have 3 wonderful professors joining us, and they will give brief presentations about their research. I'll give myself a brief intro now on each of these presenters, and after they will present and we'll come back together for questions from the audience. I will start with Professor Lydia Bourwiba. She is a physical applied mathematician concentrating on geophysical problems of hydrodynamic turbulence and on the mathematical modeling of population dynamics and disease transmission. She joined the Department of Mathematics at MIT in January 2010 as an NSERC postdoctoral fellow and lecturer. She previously worked at the Center for Disease Modeling in Toronto, Canada, on the modeling of influenza. Dr. Bourwiba completed her doctorate from McGill University, studying rotating homogeneous turbulence theoretically and numerically. She is now an assistant professor in the Department of Civil and Environmental Engineering at MIT, where she continues to focus on problems at the interface of fluid dynamics and disease transmission, with the aim of elucidating the fundamental physical mechanisms shaping the epidemiology and disease transmission dynamics in human, animal, and plant populations. She holds the Esther and Harold E. Edgerton Career Development Chair and is Associate Faculty, Institute for Medical Engineering and Science. Dr. Elsa Olivetti her research focuses on improving the environmental and economic sustainability of material flows in the context of rapidly expanding global demand. Her research spans 3 levels of materials production: operational, industrial networks, and market strategies. Dr. Olivetti's research addresses 2 major problems: improving the sustainability of materials through increased use of recycled and renewable materials, recycling-friendly material design, and intelligent waste disposition, and understanding the implications of substitution, dematerialization, and waste mining on materials market. Her research disciplines include energy and the environment, manufacturing, materials processing, and materials systems and analysis. Dr. Olivetti has a BS Engineering Science from the University of Virginia and the PhD, Material Science and Engineering from the Massachusetts Institute of Technology. As for Dr. Hazel Siv, she studies development of the vertebrate embryo. Her group has made unique contributions to understanding how the face forms and how the brain develops its normal structure. Research in the Siv lab emphasizes craniofacial and brain disorders using the accessible frog and zebrafish models. She is a pioneer in analysis of the extreme anterior domain, a unique and important embryonic region she named. A major focus of her research has been on development of the nervous system. Using novel techniques, her lab defined some of the earliest molecular markers of the nervous system. These answered the age-old question of when the embryo decides to make a nervous system and showed that future brain cells are set aside when the embryo is just a ball of cells. She also has a long-standing interest in understanding neurodevelopmental disorders, including those relating to mental health. She is a member of the Whitehead Institute, professor of biology at MIT, and associate member of the Broad Institute. Professor Siv received her undergraduate degree from the University of Witwatersrand in Johannesburg, South Africa, her PhD in 1986 from Rockefeller University, New York, and carried out postdoctoral research at the Fred Hutchinson Cancer Center in Seattle, Washington, before joining the MIT faculty in 1991. She was the first associate dean for the MIT School of Science and is founding coordinator of the MIT Africa Initiative. Most recently, she was named a 2015 MacVicar Faculty Fellow, MIT's highest teaching award. This morning, Professor Bourwiba will speak first, followed by Professor Olivetti, and completing with Professor Siv. After the three professors speak, we will open the floor to your questions. Thank you for your patient listening.
All right. Okay. Thank you for the great introduction and for the invitation for all of you to be here today. So I'll tell you a little bit today about the research that I do in my group. And I wanted to start with the slide showing you actually some of the members of the group that are contributing to the research. And the theme of research in my lab is the fluid dynamics of disease transmission. So why study infectious diseases in today's basically age and entering the 21st century? Shouldn't we have eradicated infectious diseases by now? And in fact, that was the optimism of the beginning of last century when antibiotics were introduced, improved hygiene measures were introduced, public health agencies and controls were introduced around the world. There was a major optimism at the time that we would have eradicated infectious diseases by the end of 20th century. And in fact, we had one victory in our human history in eradicating one infectious disease, which was basically smallpox. Unfortunately, we had also one other goal by the end of last century to eradicate polio, which was not achieved. And we continue talking about infectious disease eradication, like for the Surgeon General code that General Williams that is on the slide there. Unfortunately, today, as you hear in the news regularly, and with the recent outbreaks of H1N1, H5N1, and then SARS, now MERS, et cetera, infectious diseases are actually still with us and will remain with us and increase in terms of outbreaks and speed of spread worldwide in the coming century. As you can see here, actually infectious diseases, and this is not even updated, these are not even updated numbers, infectious diseases are still causing more than 25% of cases fatalities worldwide. So that means that they're at par with leading other diseases like heart disease and all of that. But one thing that in this part of the world we often don't realize so much is that one of the leading infectious diseases that kills worldwide is actually a respiratory family of respiratory diseases. So from tuberculosis to basically other infectious diseases that attack the respiratory tract. And tuberculosis was for the longest time associated with basically poor living conditions and also bad ventilation. But with the emergence also of HIV in the second part of the last century, the problem got exacerbated, and more and more we have cases now of TB, HIV coupled infections that are very difficult to eradicate. This week, there was also a meeting at the UN that actually started focusing on problems of superbugs. And what are superbugs? Superbugs are actually these pathogens that now are becoming more and more resistant to antibiotics. And this is quite concerning because, as I said, the hope that started last century was coming in part with organization of public health measures, organizations, improvement of hygiene conditions and living conditions, but also in big part because we had antibiotics that were basically emerging that allowed all sorts of infections to basically not develop or be suppressed. With today's emergence of antibiotic-resistant strains that are basically now on the rise, and very limited research in actually developing new antibiotics, we are basically now facing the potential in the second part of the century of returning to a pre-antibiotic era. And so it becomes very important to start understanding also through all sorts of angles the propagation and of infectious diseases in the world, and also not only focusing on research necessarily just on vaccine development, but all sorts of other strategies for stopping spread and mitigate the spread early on during outbreaks, even when we don't know yet the extent of the infectiousness or virulence of a given pathogen. And so this is just to give you a little bit of a summary of the gaps in the research that are ongoing. Obviously, a lot of people are looking at trying to stop diseases and curb the infectious diseases worldwide. Unfortunately, the research so far historically have been focused on the very small scale, so understanding infection at the cellular level, so looking at, for example, viruses infecting cells and trying to develop vaccines from basically microbiology perspective, and other investments in the second part of the century due to development also supercomputing have been directed toward research at modeling at the large scale, pretty much like climate models. The idea of having spread models that would give us risk assessments of how quickly something would spread worldwide if you have an outbreak somewhere in the world. Unfortunately, connecting these two scales hasn't been very much done. And the issue with that, if we're not doing that, is that we have a very hard time entering parameters and actually accounting for physical processes that would be happening, for example, at the scale of this room, in terms of transmission from one person to another or within this building, into the larger scale models of simulation that are trying to assess these risks and speed of propagation of outbreaks at more of a worldwide level. And so where my research lies is in between. What we're trying to do is sort of be at this intermediate scale to try to link the microbiology scale, be able to leverage from a mechanistic point of view the understanding of the transmission process so that we can block it at that scale and on the one hand, and on the other hand be able to also push back upscale the parameters of the small scales by knowing exactly how they would upscale in the nested type of models families, for example. Okay. Where should I point? All right. So a little bit of my background. So you heard about my background a little bit in fluid mechanics. So I worked in both fluid mechanics, so that's very much physical processes and modeling at that intermediate scale. Epidemiology modeling. So I did work in these large-scale models, and that's where my awareness of the limitations of the parameters and understanding of mechanism came about. And my group today focuses really at this intermediate scale. In terms of areas of research that we pursue, we're looking at respiratory disease transmission, and I'll talk mostly about that, but then also nosocomial infections. So these are infections that are acquired typically in the hospital that also involve contamination of surfaces or air, and then waterborne diseases. But I also wanted to branch out and show you another example of how we can also translate the approaches and the results that we're gaining from human diseases to other type of diseases, like basically plant pathogens, which also basically contain pathogens, are driven by pathogens that are transported through air and water and can affect also our food system, which in turn is coupled with the overall health of the world, because malnourished society is also a society that is more susceptible to infection. So how are our respiratory pathogens actually transferred or transmitted? And I'll show you here some of the approaches that we have, but first I want to contrast with what we understand and what are the rules or at least the regulations that are out there, for example, given by the WHO, the World Health Organization of the CDC. So you probably heard during the Ebola outbreak or even during SARS, now MERS, these concepts maybe in the media about large droplets and small droplet transmission. Typically for large droplet transmission, people are thinking about very short distances and being in very close contact. And for small droplet transmissions, which was basically what was expected for SARS, we're thinking about basically potentially longer distances, but without really knowing exactly how much. The rule of thumb in hospitals is about 1 to 2 meters, but when you actually look into the details of where is this classification coming from and what are the distances that are given coming from, you realize that actually the scientific basis for this classification comes from the work of Wells from the 1930s. And Wells did a very groundbreaking analysis at that time, but it's still from pretty much stayed at that level of analysis till now. And he said that when we are sneezing or coughing, we're emitting basically droplets. And if you consider a droplet as basically a sphere that is settling at a given settling velocity that you can compute a typical average time that this droplet will reach the ground. And if it reaches the ground faster than it would fully evaporate, if you compute that also with having an evaporation rate proportional to its surface area, then we will call it a large drop. And if it's the opposite, then we'll call it a small drop. We don't know where it will go, but we'll call it a small drop. And this is still the basis of the World Health Organization classification. So in fact, let's revisit that. If we look at, for example, so this is the type of imaging that we do directly using the legacy of Edgerton and directly visualizing, for example, virulent expirations. What you see there is basically a sneeze. That's how a sneeze looks like when you are basically slowing it down. This is at about 2,000 frames per second. And what you see there, and you're trying to identify large versus small droplets, is that in fact it's not just individual droplets that are coming out during this process. You have a continuum of droplets that are coming out. And in fact, what was revealed by this series of visualizations is that it is a multi-phase turbulent cloud that is coming out, which so far have been ignored from these descriptions. And so to which extent adding the description of a turbulent cloud that is carrying all these droplets becomes critical in assessing times and distances is what we looked at in this first phase of the work. And if we revisit here, on the left panel, you have droplets coming out for a cough. On the right, you have the superposition of the trajectories of the drops. So you can see that the larger drops could be considered almost as ballistically being emitted in terms of their trajectories and ranges. However, again, if I contrast again the sneeze that you see at the bottom panel, on the left you have the sneeze cloud, but on the right you have the highlight of the droplet trajectories, of the larger droplet trajectories coming out of this cloud as well. You see that definitely we should separate the very, very large drops from the continuum of small drops, but the small drops are not just all the same, and they're definitely not being emitted ballistically. They are carried into a multi-phase turbulent cloud that is basically moving into a room, growing, encapsulating ambient air, and then eventually leading to the settling of these drops. But the key point is that a drop of a given size is not always going at the same distance. It is the physics of the cloud and the turbulence coupled with the ambient air conditions, with moisture and temperature, that will prescribe the range of deposition and contamination of this cloud, of the droplets. And what we find is that, in fact, the droplets, so I'll skip this, the droplet transmission and range is heavily dependent on the cloud dynamics and can be extended by a factor of more than 200 with respect to what we would compute without the cloud being there. So let me just skip a little bit all of this and go to another component. I wanted to show you some experiments, but I'll skip. But basically, what we do is have direct visualizations, but then develop mathematical models, and go back in the lab, do controlled, actually, fluid mechanics experiments that we can use to validate the models. And this is also the approach that I wanted to illustrate in this talk in terms of our scientific approach to the problem, which hasn't been done and cannot usually be done in epidemiology, where we can't just run experiments. And so our hope is that we can construct contamination maps and have all sorts of additional insights that are gained in the transmission process because we're relying on the scientific approach of hypothesis, tested experiments in the lab through fluid mechanics, and then validation. And maybe in the last few minutes, I wanted to show you illustrations of droplets coming out, actually, if you zoom in closer to the mouth, which brings us to another area of research. So sorry, it's during breakfast. But this is a sneeze looked at, much closer. And what you can see here is that, in fact, it's not even just a set of well-formed droplets that are coming out. You continue having what we call the fragmentation process, or a cascade of events that lead to a stretching of the fluid, its extension, destabilization, and eventually selects the final droplet size distribution that is coming out. And so the second area of research that we're now really much focusing on is understanding the fluid mechanics of the breakup. And this process is universally found everywhere. That what's create, for example, rainfalls. If you look at jet engines, that's how you create also droplets. You aerosolize all these droplets under various very extreme conditions. It turns out that sneezes and coughs are also extreme conditions, and understanding the coupling of the airflow and the breakup process and the elasticity and that is induced by the biological processes in this fluid is critical to also understand the size distribution of these drops and what they contain and their potential virulence, to couple that in our different models. And I'll end here, and I'll invite you to look for more on the website, and I'm happy to answer questions.
So as we all get out our hand sanitizer and our napkins, I'm going to switch gears entirely here. But one thing that's been really wonderful about being at MIT this year and is all the events surrounding the campaign and the centennial is that I've had a lot of opportunities and am honored to be able to speak to groups of alumni. We always say the thing that's wonderful about MIT is the people, right? And so the energy and the inspiration that is possible for us as young faculty, as Lydia and I, is to get to interact and be inspired and energized by the contributions you guys have made. So it's wonderful to be here. There I am with my face in a laptop. So what I'd like to talk to you all about today is the work that we're doing in my group to quantify the impact of and provide strategies for improving the resource effectiveness of materials. My background is in material science. I did experimental work here for my PhD, but what I've really always been inspired by is looking at the systems impact that materials have. How do we design a material and a process at an atomistic or machine manufacturing line level? How does that relate to the system in which the material interacts, and vice versa? So it's this sort of scaling societal down to how we think about materials design at the lab bench that's really exciting to me. And in the few minutes I have with you all this morning, what I'd really like to convey is why MIT is the place to take on these problems. And there's some nuance, there's some complexity to this, and the problems that I work on invite collaboration, and this has been an awesome place to get to do that. So I won't get so much into the work that my laptop face here is going on, but we do a lot of work in electronics, and so do a lot of dismantling work, and so that's the brief explanation for why my face is in a laptop. Let's see if this will. Boop. Okay. So here, I'm Elsa. I'm in the Department of Material Science here, and these pictures are part of what motivates me to look at this. So the picture on the upper left there is the Bingham Canyon Mine in Utah. It's a Kennecott copper mine. It's one of the, I can see it when I'm flying into Salt Lake. You see this amazing structure. I've heard rumors, although I haven't confirmed myself, that you can see it from space. And then on the low, so this is about as far upstream as we can get in the extraction of copper. And then the picture on the bottom right is, you probably can recognize, bales of aluminum cans at a recycler. I think these are actually taken in Charlestown at Casella. That processes all of MIT's waste. So we like to look at these design and materials throughout the whole system. system from extraction all the way to end of life? And how can we think about how those material systems might operate more effectively to address some of the challenges that we have based on the resources that we extract and put back into the ground in landfills? So this is a not super sensitive clicker. So there are a couple of ways that I like to think about why I take this look at this problem from a materials perspective. This is data from 2009, so it's a little bit old, but you can see the reason why I like it is that it gives us a snapshot of how our carbon equivalents, so global warming potential equivalents, break down by industry and by activity. And so you can see that we're looking at electricity, heat and cooking, transportation, agriculture and forestry, so that's all our cows down there at the bottom. And then the blue bar is the materials and manufacturing, exclusive electricity, right? Because we put that in a different piece of the pie. And so that's about a third of our global carbon equivalents. And so materials and manufacturing processes is a real opportunity. And this is what I use to get my students excited about it. We have a real opportunity to address it if we look at things from a materials perspective. And then I break that down by material type. And there's two important aspects of this. One is that obviously our impact is dominated by the materials that we use in the greatest volumes, right? Steel, cement. Not maybe the most sexy of materials, but there's lots of opportunities there, and there's renewed interest in addressing those. So that's an important piece. Look at where the volumes are. But there's also this pretty big other part of the pie, right? And so that's where we get our precious metals and some of the rare earths. And so there's opportunities on both sides of that. My transitions are over there. So that's about the mass, the volumes that we have to work with. And then I like to juxtapose that with the complexities of our systems. And maybe some of you have seen these pictures before. But what we're looking at here are 3 snapshots of the periodic table, where we're looking at the elements that are used in circuit boards over time. So in the 1980s, we've got about 11 elements. Then in the 90s, we added about 4. 2000s added about 45. And what I say now, if I had my phone up here with me, is now you basically carry the periodic table with you around in your phone. So I'm sure you guys have heard that sort, but it's basically true. And so this juxtaposition of these large quantities with this increased complexity means we have a bit of a disconnect if we couple that with how we've been doing in recovery of those materials. So I think this is my last view of the periodic table. But what this is showing is how much of those metals are we recovering? So this is a metals-focused look. So I showed you we're using everything. Here's how we're doing at recovering those things at end of life. So we've got the pink is the worst, sort of less than 1%, up to blue, which is greater than 50%. But only about half of those that are greater than 50% do we consistently recycle at that level. So we've got a large volume, we've got a lot of complexity, and we're not so great at getting them back. So this is why I've got a whole host of problems that we like to look at in my group. And what I'm going to tell you are just 2 little snapshots that get at what I was talking about before, about how these problems are complicated, even if we're looking at pretty simple systems, what we would think at pretty simple systems. So this is a picture of an alkaline battery. Very simple. We've got zinc and manganese-based system in a steel can. The industry has worked really hard to get contaminants out, no more mercury by manufacturers in the US anymore. So what the industry likes to say is they've worked really hard to make this product a lot like dirt. So what we did, a few years ago, California put a ban on putting these products into landfills. And so then the industry wanted to look in what were the impacts of the recycling technologies that were available at the time. And based on the fact that there were not very many recycling technologies available in the United States, and that those technologies hadn't been designed for alkaline battery recycling, so they weren't having a very high recovery, we showed through our work that it was significantly environmentally better to throw these in the trash than to recycle them. And that doesn't sit right with us, right? It's got steel. I'm sure we all have drawers full of batteries, right? Because we don't know what to do with them. And so, so there was a lot of modeling that went into understanding what that impact was. We went to the facilities that were recycling them. What were they recovering? To understand what those impacts might be. And because we learned that really this ban didn't make sense if they were trying to improve the environmental performance of these products. And what that led us to is what were the alternatives? So we were able to identify a few lower energy recycling technologies that could be developed. Even maybe more a quick fix, so to speak, was leveraging the steel infrastructure, electric arc furnace. You guys maybe have heard them as mini mills. So if these batteries could be, which are distributed throughout the United States. So if instead we could find ways to incorporate those batteries into into mini mills throughout the US, then it did make environmental sense. But that wasn't where the battery industry was thinking and what they wouldn't drive at. So that's just an example. So you should still throw your batteries in the trash because they haven't quite got that sorted out yet, but, but they're working on it. So the other quick example that I want to give you sort of feels like a basic sort of run-of-the-mill kind of problem, but something that takes some detail to look into is the recycling of this object right here. So what you're looking at is a Sankey flow, or a flow of materials in the US for paper and paperboard in 2003. And we obviously can't go through all the details in here, but I put this up here so you can see the complexity of the way this system moves, right? So we've got those yellow strips in the middle. Those are the paper and paperboard products, so cardboard, toilet paper, tissue, anything. And then the blue flows are the virgin flows, so the flows of virgin trees that we have going into those products. Oh, sorry, I said that backwards. The brown is the virgin flows, and the blue is the recovered flows of material. And then you see that going back around. So if I could make this round, the blue bars would match on the other side. So the trick with this work is that there's a lot of, the policies that we have around paper recycling in the US are written around this product. They're written around putting recycled content into this product. But you see all these flows are connected. So as we're putting our recycled content into this product, and if we don't have enough recovery, if it doesn't also increase the amount of recovery, what we're doing is putting virgin into things like tissue and toilet paper. And so there's this, at least in the US right now, there's this kind of perversion in the way that the flows are moving. And so these policies need to be designed in the context of the technical constraints of the mills that are managing this material as it comes in, and also the municipal solid recycling facilities that are recovering this paper and bringing it back into the system. So these are just two illustrations of the fact that even things like alkaline batteries and paper, there's still a lot to understand if we're actually going to move the needle on the impact that our materials have. So with my last few minutes here, I'm going to tell you a little story of a technology development that we've been working on. So this is a little bit more sort of focused on taking waste materials and trying to derive some value from them. So through the Tata Center for Technology and Design here at MIT, that some of you might have had some exposure to, we've been working with a set of industrial facilities that make paper, sugar, steel, bricks in northern India, at a town called Muzaffarnagar. And so we've got India there. It's in Uttar Pradesh is where we're studying. We zoom right into Muzaffarnagar. And then what I'm showing you here is a GIS map where we've mapped all the different factories in the region. Mostly what you can see there are these brick kilns that are scattered all around, these green circles. And then I don't know if you can see, but in the bottom right there are a series of black triangles. Those are the paper mills. So the challenge that this region of industries was facing is that they had a lot of environmental waste, both emissions to air. They were dumping black liquor, for example, from the paper mills into the rivers because they didn't have another option. These were all small and medium enterprises. They still are. And so what they needed was a low-cost approach to managing this waste, because that was the only way they were going to actually manage it responsibly, because they didn't have the money to do so otherwise. And so in particular, what we helped them with was looking at waste boiler ash that's produced in the factories. So anything they can get their hands on that has calorific value, they're going to burn for energy production. And in cases where there's incomplete combustion, they're left with a lot of boiler ash. So you see these materials on the left, pet coke, rice husk ash, bagasse, sometimes tires, going into a boiler to produce energy. That's the green arrow. And then the waste is going to landfill. And there's a significant amount of this ash in the region. They have to buy these plots of land to just dump the ash on these landfills. And they're compensating the people that live around these for the particulate matter that they're getting in their eyes. So we've got this significant waste issue that they need an economically feasible way to manage. We juxtapose that with the fact that there's also this increased need for housing in the area because of the increased population. And you saw those little green circles of those brick kilns. All of those brick kilns are clay-fired, running at 100 degrees C to make bricks for the people in the region. And they're excavating the clay, the topsoil, in order to do that. So there's this housing demand, there's the challenge of the waste, and then there's this issue with the impact of the clay excavation on the area. And so what we did was we made a brick from the ash. So essentially, through alkali activation, so sodium hydroxide activation of the aluminosilicate component, chemical component of the ash, we're able to cure a brick at 30 degrees C compared to the sinter temperatures of the clay-fired system. So the traditional clay-fired brick is there on the up above. You guys have all seen red bricks before. And then our brick is the one at the bottom at 30 degrees C. And you can see it's 70% by weight ash in our brick. So that helps with taking care of that volume. And then we've added some clay and some lime and some sodium hydroxide. And then you just see an example of the kinds of results we have there. So on the bottom right, you see ash from 3 different paper mills. We wanted to demonstrate that despite all the fact that they have all this variety coming into the mill, we can still make this product. And the dotted line there you see is the standard for brick strength for those clay-fired bricks in India, 7.5 megapascals. And so even after only 3 days at 30 C, this chemistry is competitive, is on par with that. With the bricks. I don't know if you can see the plot on the lower left, is that we can increase that strength significantly if we increase the compressive strength of how those bricks are being made. So just something that we're able to play with in the field. All this needs to be done in the field, right? That's the issue we're trying to resolve. And I always get lots of great questions about the health effects, and I'm happy to talk about that after if we have time. What I will say is we've done a pretty detailed environmental and economic analysis of this product, and it's environmentally much improved over the clay-fired brick and economically on par. And so what we've been doing next with this work is prototyping and using our bricks in the region. So my research team was back in India. They go twice a year. So they were there in the summer, and they made about 500 of our bricks sort of in the field, at production, and built this little structure, which I feel looks a little bit too much like an outhouse. But the point was to illustrate that we could make the bricks at some kind of scale, 500 bricks, that they would be consistent, and that we're able to make corners and that sort of thing. So this was about a couple months ago. Our partners were here last week for a symposium, and they brought us a picture of the wall. It's still there. It's still standing. And it's been one other thing that's been really great about that project is being able to partner with the local industry to work with them on how this product would be implemented in the area. So thank you all so much for your attention, and I'm looking forward to your questions in the moment.
Okay, welcome. It's a real pleasure and honor always to speak with alumni. Welcome back. Your role in the Institute and keeping this place great is really immeasurable. We're an interesting trio, my colleagues and I. I'm kind of trying to think how to put it all together. There was some biology in the beginning, and it had something to do with your face. So that sort of, interfaces with what, so to speak, with what I'm going to tell you here. And then, Professor Olivetti talked about, juxtaposing materials and so on. And so we could kind of go there a little bit with what I'm going to tell you. But really what I want to tell you is about a project that I love. I've been working on this project for, a couple of decades now. And as you'll see, and we can discuss in the Q&A, there's some real medical implications to this project. But I thought I would tell you something cool rather than trying to go for the pragmatic, which my colleagues have done so well. Point it to look, it worked. Okay. The way to get through a bad day for me is to think about that once, I was a single cell and now I'm me, and even if it's not such a great day, that is a pretty awesome trajectory. So I pass this on, as a strategy here, because it really is truly fantastic. As an undergraduate, I started looking at frog eggs and following them over a period of days and seeing them turn into tadpoles and ultimately frogs. And I thought, wow, how do they all know what to do? How do 10,000, 100,000 embryos that were all fertilized at the same time, go synchronously through their development, all become tadpoles at the same time, all become little frogs at the same time? How do people develop? Where did I come from, et cetera? It was really a fantastic spiritual experience, really, and it led me down the path of asking what molecules were involved in telling these eggs and embryos what to do. And that turns out to be an extraordinarily rich field that has everything to do with normal development. It has a huge amount, of course, to do with abnormal development and birth defects. It has almost everything to do with cancer. It has everything to do with using stem cells to regenerate organs. All of this came from and still comes from understanding how development works. The idea is that you go, no, I didn't do anything there. The idea is that, as I say, you go from the single cell to the complex animal or plant, where there are millions of cells, lots, hundreds of different kinds of cells, and all of these are arranged in the 3D structure that makes us so attractive and functional. The part of the embryo that we have studied for a long time and that I'll talk to you about today is the face. That defines us as who we are, that allows us to communicate, and apropos our gathering now, allows us to eat. We study the tadpole, we study the frog. Frogs are very accessible. You can get lots and lots of embryos. They have very nice flat faces, so you can see what's going on. And that is our model, which is why I'm showing you the tadpole face. And in particular, we're going to talk about the mouth. But let's just consider the whole face for the moment. There is an extraordinary kind of dance, movement, interaction between the cells that build your face. In this animation, the different colors are different groups of cells, each of them hundreds of thousands of cells building the human face, and the black in the middle is the mouth. How do these cells know what to do? How do they know what to build? How do they know how to move? Who are those cells, in fact? These are the kinds of questions that we've been asking for some years. And the part of the face that really we've focused most on is the mouth. So I'll talk to you a little bit about the mouth, and then I'll talk to you about something called reciprocal signaling, which kind of sounds material science-y, and it is in a biological way. Okay, so the mouth. Let's talk about the mouth. When we think of, an ancient face, really it's the mouth that we're talking about. As soon as animals in the evolutionary record became multicellular, they got a hollow cavity in the middle. Inside that hollow cavity was the digestive system, and it was the challenge for the animal to get food into the digestive system, so you needed a hole that was the mouth. The mouth is present in every multicellular hollow animal, which is pretty much every multicellular animal. You can see in the sea anemone there's a beautiful mouth in the middle, and in the earthworm, the little face of the earthworm, so sweet with its mouth, okay, kind of almost prototypical faces, okay. From work and research we've done, we think that the mouth evolved once, and that the way our mouth forms is very much the same as the way the sea anemone mouth formed, and the way the earthworm mouth forms, and the way the sea urchin mouth forms. We also think about the mouth as what we call a scheduled hole. So, the embryo, and indeed the adult, has got a protective covering, and if it's punctured, you have a wound, and that's a bad thing. The mouth, in fact, is a hole that breaches the protective covering. And that could be a bad thing too, except that it's not, because the hole is scheduled. It's organized to form at the right time in the right place, such that it connects with the digestive system and makes a productive opening rather than a wound. And there are a listing of several of our holes that all form in this kind of scheduled way. And in another time, I will tell you about the notion of the biology of holes that we've been playing around with. Okay. As I said, we think there was a mouth that arose once during evolution, and that process has been maintained throughout the evolutionary record. In fact, you can see that because in all animals that we've looked at, the mouth arises from a particular part of the embryo where those blue cells and those yellow cells are juxtaposed. We call that the extreme anterior domain. And we've studied this in frogs, but there's an exact same domain in human embryos. So how do you make this scheduled hole of the mouth? Well, you do it like all developmental processes. You know, you don't go from the egg to the frog in one easy step. You go from the egg to a ball of cells, and then other stuff happens, hundreds and thousands of steps to get you there. Same kind of thing with the mouth. In the mouth, you have those blue cells and those yellow cells juxtaposed to one another. There's a layer called a basement membrane between them, separates them. It disappears in a very controlled way, just in the region that's going to form the mouth. The blue and the yellow cells thin out. Some of them die, some of them move away, and then they intercalate with one another, and they thin and they thin and they thin until eventually a hole is made. This is a side view, and these are steps that we've described here at MIT. If you look from the front, you can see the developing tadpole, and this extreme anterior domain that's going to form the mouth I've outlined in red. And you can see that as the tadpole develops, the shape changes of that region, and eventually it opens up into the mouth. And we can do some diagrammatic depictions of all of the various events we've studied, all of which are associated with particular genes, and I'll tell you about one in the end, and show that the blue group of cells, we can't see the yellow cells, now we can just see the blue cells, which are going to be the outside part of your mouth, the lips, and the lining of your whole mouth, and some of your teeth. We see those cells start off as a big kind of square, and then the cells are told to organize themselves into something I wrote as a pre-mouth array, which is a long, thin layer of cells. And this is there really, really, really early during development. And it sits there as a kind of a line of cells waiting to be told that it can open down the middle. And when it gets the signal, and we don't know what the signal is yet, it starts to open up and unzipper, and you get the mouth opening being made. Okay? So we claymated this, and we also did some microscopy. You can see the green is the microscopy, looking right head-on at the animal. And the claymation is obvious, and you can see how when we claymated this, you can see what those blue cells are doing. You can see how they started, maybe, ooh, this is like such an independent type of, hey, I didn't know what Elsa was talking about, but it's like this, you know? There's a relationship that you have to form with this clicker, right? Huh? There we go. Okay, there's the cells, you know, getting long and thin somehow, and then they split down the middle, boom, and your mouth opens, okay? It's pretty awesome. This is the first claymation of mouth formation, and we were really excited to do this. Okay, so, so how does this all work? You know, how do these cells know what to do? And they know what to do because of the genes that are active and telling them what to do, but that's a kind of trite answer. So I'll try to be a little more specific and tell you that there's a process of signaling, chemical signaling, that is a conversation between two groups of cells that form the mouth and that form the rest of the face. So if you look at my diagrams of the face here, the outline of the mouth is in that red box. And then you can see there's this green color that's creeping in to surround the red box. Those are cells called neural crest cells, and they're going to build all the rest of your face. The jaws, the musculature, the nerves, and the blood vessels, and so on. And those neural crest cells have to know what to do, and they have to know where to go. And what we discovered is that way before there is a mouth, when there's just a group of the square box of cells, that group of cells, the extreme anterior domain, is sending out signals and attracting the green neural crest cells into the face. Okay? So it's acting as a beacon, a kind of signaling beacon that's getting those neural crest cells into the face. And we had no idea that this was how it worked. So this was a really exciting thing we found in the last few years. We found this by using a technique we developed of facial transplants that you can do in the frog, where we can take little pieces of tissue, just this extreme anterior domain, from a normal embryo or from one that's lost the function of a particular gene of interest, and transplanting that into a recipient embryo. And you can see in the tadpoles, the little green area is the transplants, just a tiny group of cells in an otherwise normal embryo. And when you have an abnormal extreme anterior domain, you see that the face is really abnormal. There's no mouth, and everything does not look correct. What we also found is that the signal that is attracting those neural crest cells into the face is the same signal that your blood pressure medication targets if you're taking it, or if you know someone who's taking blood pressure medication. And we didn't know that either, that the embryo was using a signaling pathway that is used much later throughout our lives to control the diameter of blood vessels and our blood pressure. And so that, just throwing out there, if one is pregnant, it's not a great idea to be taking blood pressure medication because that would harm the face of the baby. And then, a last piece of data that I'll show you is that we can actually show that the signaling pathway, called the bradykinin pathway, is really required for face formation. We can knock out the genes involved in this pathway. There on the left-hand side, panels E and F, you can see very sad, thin little faces with no mouth and no neural crest, no jaws. And we can make those embryos better by taking a little bead, a synthetic bead, soaking it in the peptides, the bradykinin peptides that this pathway makes, and then implanting them into the embryo, and like magic, you see the mouth coming back, you see the whole face coming back, the jaws, the musculature, and so on. So we can show that this pathway is really one that's got tremendous power in building the mouth and building the face. Okay, and if that was just like whatever, we can also do art with the facial transplant. Okay. So I was going to, whoa. There was something else I was going to tell you, but actually, I tell you because maybe I got 1 second. I don't know where it went. Somewhere else. Okay, I'll just tell you while you're looking at the art that actually the reciprocal signaling has to do with the cells that come in from the neural crest. Once that mouth region has attracted the neural crest then tells the mouth, the region that's going to become the mouth, to continue its trajectory and actually open up into a mouth. And that was the reciprocal signaling that we zipped through a little quickly, but there we are. So there is a whole host of people who have worked on this project. I think we could not really have done it anywhere except MIT, where anything is possible. So thank you very much for your attention.
We will now open the floor for questions from the audience. Yes, can we have yes, right there, please. Here. Can you, the gentleman here, please.
I was interested to see the infectious diseases in North America and how many there were compared to the tropical areas in Africa and South America. Is that just because we study them more, or are we really rife with infectious diseases here?
Can you still hear me with this? Yeah, okay, great. So in the map basically included all sorts of emerging and returning diseases that have been identified or recently or re-identified. But definitely there have been, unfortunately, more emphasis on studying diseases that affect the developed world, and that's the reality as well. Even though there are more and more initiatives now with foundations, etc., to look at infectious diseases that are touching the developing world, it's still not at par with the investments in diseases that affect the developed world.
Next question. Yes, you have. Can you get the lady there, the mic?
Do we have any idea of what the relationship between TB and hygiene is?
So I'm going to give you a sad anecdote. But so I'm from Canada, and in fact, Canada has a rate of TB infection that is at par with the developed world, except in the native reservations, where actually the numbers are comparable to the developing world. And if you go to the native reservations, there's definitely a problem in terms of the housing, the facilities, the infrastructure. So definitely TB is more prone to be transmitted and to persist in environments in which you have very poor ventilation, overcrowding, and also poor immune conditions. So if you're malnourished or the health is not optimal. The big issue nowadays for TB, however, worldwide, is the fact that we have that coupling, for example, in Africa, particularly with HIV. And in addition, now with the superbugs, so the fact that we are having a hard time controlling it with the antibiotics that we have, that is a pretty scary prospect that would be independent of the environmental conditions, even though it's still exacerbated by it. But yeah.
Yes. Okay. Okay, go ahead, sir.
Good morning. Dr. Olivetti, I liked your presentation very much. I did my master's thesis on remanufacturing technology here. Do you also look at the repurposing of materials instead of bringing them down to raw materials? In particular, I work for GE, and we're going to have thousands and tens of thousands of windmill blades that need to be either recycled or repurposed or put into landfills. So that'd be an interesting project for your team to look at.
No, that sounds great. I've actually always been really impressed. GE has a great department there, so we've had some collaborators there that look a lot at those kinds of issues. So yes, I mean, a lot of what we do is, given a set of technologies or potential technologies for refurbishing, remanufacturing, or recycling, what makes the most sense given the context? I mean, I always say that the answer to every question I've ever asked, maybe these guys would agree, is it depends, right? And so our goal as in the group is to understand under what contexts we can be confident that we're making this choice correctly, given the uncertainty, given the data, given that sort of thing. So it's definitely part of a set of strategies. I think probably one of the repurposing examples that's come up the most recently has to do with electric vehicles, and that you have, in an electric vehicle, you've got a pack of battery cells, essentially, that you could repurpose once the battery is done with first life into smaller batteries that could be used in electric bicycles, that sort of thing. So that's an example that's become recently of interest. Thanks for the question.
Next question.
A quick follow-up, too. Have you also the eco-design directives in Europe and the DOE provide some interesting guidelines? Is that something the students are becoming aware of, too?
Yes. I mean, particularly if there's someone in the audience, I would love some good, strong examples of design for the environment. We've got lots of sort of pieces of examples, but the sort of strong example where there has been a good connection between the designers and the folks at end of life. We do a lot of sort of theoretical pieces of that, but given the system and the context that things evolve, I think it's hard for those sorts of things to become context. So maybe we'll follow up after.
Next question.
Dr. Siv, I'd like to know applications for humans in terms of the formation of the mouth. What I'm thinking about, for instance, is cleft palate. Is there any applications of your work in terms of prevention for cleft palate?
Right. So I think, the mantra is that we have to understand how it works before we can fix it, okay? And I think that to some extent that is true. I think what the understanding that we have personally got is that cleft palate often comes together with a whole bunch of other things. My cousin runs a genetic testing program down in Philadelphia, and when I was telling her about my work, she said, "Wow, your frogs look just like my patients." And we met the patients, and in fact, they look just like the frogs. There's a whole bunch of things wrong with the face. And so we think it's this particular region of the embryo that we've looked at that is directing a whole bunch of things, and bradykinin and some other molecules involved. So, you can go somewhere with that to say, okay, maybe we can actually give those compounds at some point if we can detect very early during development that there's something wrong. Or maybe we can later on go and take the right kinds of cells, the right kind of stem cells that we know from working with the normal embryo, and maybe we can repopulate cells, the clefted palate and the parts of the face that are abnormal after birth. Cleft palate treatment now involves 15 sequential surgeries over time because the implants are really static and they don't grow. And so the real goal is to put in implants that would grow and change with the face and correct the defects, right at the outset. And so absolutely starting with, the embryo, understanding the embryo, and then marrying together bioengineering and basic biology and, chemical signaling is the way to go.
Okay, last question, please. Can we get a mic over here, please?
Professor Burry, a question. Looking at your slide of initially all the pathogen, isn't there kind of a conservation theorem here that pathogens, and maybe due to mutations, will continue to evolve at about the same rate you can identify them and treat them? I mean, some of these things are, you have Ebola, Zika, dengue, a few others have been around for a while. They've been known, but they then tend to have a fluorescence, it would seem. In other words, do we know more pathogens now and diseases than we did 50 years ago? Is the number constantly increasing, and why is it constantly increasing?
So, there isn't a clear answer to that, but one thing is clear is that there is more humans on the planet and less actually green areas. So we actually push the frontiers of where we live, and by getting into also areas where we didn't used to go, we are exposing ourselves to new pathogens that might be there, have been there for a very long time in other species, and the spillover issue is dominant. So spillover is when you have pathogens in other species, so animal species, for example, jumping into human species. And that's the major issue why we have a very hard time eradicating diseases. So for smallpox, for example, that was really a human pathogens, and with all the efforts put in place, we were able to eradicate it. But if the pathogen is circulating silently without any symptomatology in other species, and we don't know which ones, and we don't know how to basically target that, then a new encounter with that species would lead potentially to new outbreaks. So that's one of the main reasons why we have been having more and more of these exotic pathogens. And also, for example, influenza was timed really to emerge in human populations when agriculture started, because it's circulating in avian species, so birds, and typically a mixture of birds with water ponds, because it can survive in the water, with pig populations. You have a perfect mixer there for having the virus jump from host to host and adapt and acquire new ways of infecting humans. So that's a big part of the issue. The other big part of the issue is that we are now also very connected. So also the social organization of the world. It used to be that we could potentially quarantine, for example, for Ebola, they have had very ancestral means to stop it. The villages would just stop connecting with each other until the outbreak would go away. Today, if the symptoms are not basically obvious, we can travel and be on the other side of the world, land in New York, and have a whole outbreak, and give many more opportunities for the pathogen to adapt. So there is no clear answer of if we actually have more, or if we're just more exposing ourselves to it and exacerbating that process. But ultimately what is happening is that we can have much bigger impact, outbreaks with much bigger impact much faster today than before.
Well, thank you very much, professors, for your top-notch research and for your inspiring presentations.